Today we have a special guest, Dr. Maria de Miguel, medical oncologist and clinical investigator at START Madrid, HM Sanchinarro. You are welcome.
Thank you very much. It's my pleasure to be here with you today.
Thank you for joining us today again. We have an interesting topic, antibody-drug conjugates, pioneering topic for cancer treatment, offering targeted therapies. Well, we may start with some information about your professional journey and your background, if you share some detail with us.
Sure. So I have practically my entire professional career has been dedicated to the development of early phase clinical trials. I am primarily focused on thoracic and GI tumors, which are both my passion. And so I have been involved in hundreds of different phase 1 trials. Currently, I am a senior investigator at START Madrid HM Sanchinarro, and I'm also the associate director there, serving as principal investigator for several phase 1 clinical trials, including the new generation ADCs you were talking about.
Can you provide an overview of your research on telosuzumab aducanumab, ABBV-400 and its potential as c-Met directed antibody-drug conjugates for non-small cell lung cancer? What makes it stand out to compare other ADC in development?
Yeah, the c-Met signaling pathway is associated to an aggressive behavior of cancer cells, we know, and represents also a negative prognostic factor in non-small cell lung cancer. So targeting c-Met has become an area of interest in general for therapeutic intervention, as it is overexpressed in an important rate of these patients. In this setting, telosuzumab aducanumab, it's a new ADC that's targeting c-Met, and it's conjugated to a topoisomerase 1 inhibitor payload. And as we are going to see, it is showing some exciting preliminary efficacy. Things that are different of this drug is that this is a novel payload with the topoisomerase 1 inhibitor, as well as it has a high DAR, drug-to-antibody ratio of 6, which are possibly the main differential characteristics of this ABBV-400.
Confirmed overall response rate for ABBV-400 in non-small lung cancer was about 43%, and the clinical benefit rate was about 85%. This is very interesting and high rate. Can you elaborate on how these results compare to other existing treatments for these patient groups?
Yeah, that's totally right. In my opinion, these are very promising and exciting results, as then the tumoral responses have been observed across different MET expression levels. Of the treatments that we have been exploring targeting MET overexpression, such as monoclonal antibodies, have shown just modest activity so far, And possibly the main competitor here to this drug is another ADC, which is Telisov. However, there are very relevant differences between one and the other. While Telisov is a microtubule inhibitor and that is lower, importantly, the overall response rate that was shown in the Luminosity trial was in general lower.
And what insights have you gained from the ongoing biomarker analysis exploring the association between c-Met protein expression and the treatment response? How might these results guide future clinical decision making?
Yeah, what we have seen so far is that the higher expression of MET, the higher response rate we get. So almost 80% responses were observed in patients with more than 50% of cells 3+. However, it is very interesting to see also responses in patients with a lower expression. Why is this happening? It might be two different facts. Maybe MET tumor heterogeneity, bystander effects, or maybe upregulation after having other treatments of conventional treatments. So what it is important in the future, even though we have seen responses in different MET levels, is to being able to establish a threshold or maybe other biomarkers to predict the niche of patients that can achieve a major benefit with the treatment.
Mm-hmm. And could you discuss the most common treatments emergent adverse events, particularly in anemia and neutropenia in patients treated with the ABBV-400? How are these managed clinically to ensure patient safety while maintaining efficacy?
Yeah, the most common toxicities, as you were saying, were hematological. Almost 20% of patients developed neutropenia and 25% of patients anemia that were grade 3 or higher. And this led to 13% of patients that require dose reduction because of this hematological toxicity. However, patients during this trial could receive G-CSF or transfusions at investigator's discretion. And with this, the patients in general had a good tolerability overall. Other toxicities that we have been seeing include GI symptoms such as nausea, vomiting, or anorexia, as well as some cases of pneumonitis.
With increasing interest in ADC for solid tumors, what do you think are the key factors that will influence the future success of the ADC in oncology? Are there any specific foresee for widespread clinical adaptation?
ADCs are a promising class of therapies in oncology, and they have become part of our daily clinics. Even in first-line treatment, they are approved already. Their future success will depend on different factors. On maybe one of those will be improving target specificity, but also trying to reduce the toxicity profile and overcoming challenges like tumor resistance or determining potential treatment combinations. Understanding whether resistance to these treatments comes from one issue or the other, from target downregulation, but also efflux pump upregulation or drug metabolism, will be key not only to know the drugs that work well, but also to establish the better plan of treatment, the best sequence of treatments for our patients. And so advances in this area will be key to unlocking their full potential across a wider range of tumors.
After presenting your finding at ESMO 2024, what key message would you like to convey to oncologists and researchers about the future of the ADC in lung cancer treatment?
This is huge. ADCs represent a powerful new weapon in cancer and in lung cancer as well, particularly in the field of non-targetable mutations in lung cancer. There are currently very few treatment options after the first line that really work, and we need some innovation here. And this is starting to happen. No? ABBV-400 and others are starting to show that the paradigm of ADCs might change this field in the future.
I finished my questions, but I don't know, do you like to underline any point or give some more information about the future of the ADC?
Yeah, as I was mentioning, this is just starting. There are many trials ongoing in many different tumor types and indications, and this is becoming the new tsunami of changing practice that we are going to see. And so this is becoming very, very exciting.
Dr. Maria de Miguel, thank you very much to be in this session with us, for sharing your insight and expertise with us. Thank you very much.
Thank you very much.